Speaker, we have Ajit Chande. Dr. Ajit Chande is an associate professor in the Department of Biological Sciences at the ISR Bhopal. His lab works on virus biology and genome editing, including studies of host retrovirus interactions, use of circular RNAs in infection, and the development of gene delivery vector. He has received prestigious awards such as the Ramanujam Fellowship, DBT Wellcome Trust Fellowship, as well as recognition as an EMBO Global Investigator. Today, he'll be talking on from HIV biology to lentiviral vectors, insights into the mechanism of antiviral actions.
All right. So at the outset, I'd like to thank the organizers for giving me this opportunity and the chair for generously introducing me. So my lab, as everyone probably now know, works on retroviruses, and we try to understand how these retroviruses evolve from their ancestors and became more and more pathogenic, and they also learn to infect various host species in turn. And while doing so, they also some of them try to jump the species barrier and cross that species barrier, and most often it was okay, but for some of the transmission events, or what we call zoonotic event, that led to the emergence of pandemic strains like HIV. With the current disease burden, almost 40 million people living with HIV, and the vaccine seems to be still far-fetched for this particular virus. Now, we try to understand in the lab that what makes this virus so successful as a pathogen and sustaining the pandemic as opposed to the other viruses, other retroviruses. So if you look at the genome of these retroviruses, what you will notice that this genome comprises of canonical retroviral genes, GAG, POL, and ANT, and these are the simple counterparts or the cousins of complex retroviruses, which without changing the sheer genome size learn and evolve to encode more and more proteins on top of what canonical proteins retroviruses encode. Now, over the years, it turns out that before 15-20 years, when people tried to study these accessory proteins, it turns out that these accessory proteins are not required for virus to replicate, right? So if you use HEK293T as a model system to produce the virus, which is defective of these accessory proteins, virus replicate fine. But it turns out the natural infection virus keeps these proteins under selective pressure, suggesting that these proteins are indeed required for pathogenicity, and the pandemic nature of these viruses is in part defined by these accessory proteins. So over the last 10-15 years, we and others tried to understand the role of accessory proteins in HIV and retroviral replication. It turns out that we have intricate innate immune mechanisms, what we call restriction factors or intrinsic antiviral factors, that can keep the virus replication in check, and these are very specific in a sense that if retrovirus is reverse transcribing its genome, there are proteins like SAMHD1 and ApoBec can keep that in check, and obviously pandemic viruses like HIV has VPX and VIF to antagonize such restrictions. There are factors that can even prevent the release from the infected cell, and there are factors like VPU that can antagonize this restriction by tethering for various enveloped viruses, including retroviruses. That was identified by Paul Benish's group and Mike Malim's group. Our lab has been interested in understanding the entry step of viruses, retroviruses, and we contributed by discovering this serine family of restriction factors that is antagonized by NEF protein from primate lentiviruses. And most recently, we identified a mechanism by which a circular RNA being hijacked by the HIV to antagonize the factor that otherwise inhibits the transcription of the virus. So in summary, that these pandemic viruses have the arsenal of these accessory proteins which they can use to antagonize the intricate antiviral mechanisms that otherwise inhibit the transmission and cross-species transmission of these viruses. Now, how this is relevant for this particular audience, so I'll just switch gears, that because this pathogen has been now converted into a useful tool, and which for 20 years ago it was never thought that this will be in clinic and curing diseases, but now we know it's a reality, that this pathogen, the genome which encodes for these accessory proteins and the pathogenicity determinants have now been replaced with the therapeutic gene cassette or the transgene cassette, and such viral vectors can enter the target cell, integrate the transgene cassette and enable the stable expression without multiplying or without causing any disease symptoms. So, having said that, these vectors then therefore will be vulnerable to the antiviral factors that are present in the target cells, usually immune cells that are being the targets for this cell and gene therapy. So, it makes a beautiful model to study what are the antiviral factors and mechanisms that exist in the these cell types. So, because they demonstrate increased sensitivity to the antiviral mechanism that otherwise exists. So, how do we identify or study such factors or mechanism? In our lab, we use something called chemical genetics as an approach, wherein we use small molecules that are known to have specific effects, or targets are known, or the pathways that they target are known. So, we develop the reporter cells, and these are seeded in 96-well format, and then we challenge them with compounds, and we add lentiviral vectors that has the reporters, and after 48 or 72 hours of transduction, we analyze millions of events using high content screening platform, and we identified situations where these are inhibited, or these are elevated, or there is no change, and we score such events and identify the candidates. Those which are enhancing probably are the ones that are useful and relevant for these clinical applications of lentiviral vectors, and those which are inhibiting are perhaps the dependency, host dependency factor that are very interesting to otherwise study from the fundamental point of view. So, one such exercise that we performed in the lab using two different heterologous envelope. One is Chandipura virus envelope glycoprotein, and the routinely used envelope, that is VSV glycoprotein. So, two such serotyped lentiviral vectors were used in the screen, and we took about 1300 small molecules and identified the events that were increasing the transduction without changing the viral titer. And such exercise identified 9 such compounds which, irrespective of the serotype or the envelope glycoprotein used, was enhancing the transduction in the target cell. Now, why two different envelopes were used? Because although these belong to the same family of viruses, that is rhabdoviruses, Chandipura virus uses a different receptor which is not yet known, and VSVG uses the LDL receptor. So, we wanted these events to be independent of the envelope glycoprotein that is being used here. So, we performed subsequent screen that included the criteria like cell viability, because in these conditions these compounds can often become toxic also, and as you can see, some of them are really toxic to the cells. And on top of that, we added additional criteria. On top of these two envelopes that we used in the primary screen, we added the native envelope, that is HIV1 envelope, to understand and to make the events only post entry, in a manner that the receptor tropism and all those aspects should not be affected by these compounds. And we found interestingly one compound that was invariably increasing the transduction efficiency. As you can just to note here that the viral inoculum is the same, and this virus is infected in these cell types, and this way you see that there is dilutions. So, in the absence of compound, this is the infectivity that you can see as scored as the green positive cells, and you can see the compound that is increasing, and even tenfold dilution of the compound, you can see significant number of cells still infected, where this can be one of the way by which the cost of production of the viruses can be reduced, because you are using 10 times less virus for getting the same number of infectivity. We check this is happening in the primary cells, and then cells are relevant, cell types that are relevant for the therapeutic applications and fundamental studies like PBMC. We see that the enhancement is there. The CD4 cells enriched from PBMCs and CD14 cells also we checked, and we are open to join the hands and test in more relevant cell types if you are interested in understanding these aspects. What we were more curious about is the mechanism by which this compound is working. So if you go back to the literature, what you find that this compound is known as FGFR1 inhibitor, FGFR1 kinase inhibitor, and several companies sell this commercially as a FGFR1 inhibitor. So we were very much convinced that FGFR1 is our target. Just to our surprise that when we knocked down all the FGFR1, 2 and 3 and 4, that 4 paralogs of FGFRs are there. So we did not see any difference in terms of enhancement. The compound was still enhancing the transduction in this target cell type, suggesting that FGFR is not the target that is very well characterized for this compound. So we went back and again did structure-based analysis where we did docking studies to understand what could be the potential target, and using the structure of this compound, we identified there are several targets with the highest probability. We found EGFR, SRC and FGFR1. Obviously FGFR1 was already known. That means that analysis was consistent with what the compound does. But it was too many to screen, so I asked my student, because this compound should not be working in anything and every cell type, right? So I told him, and the PhD student, brilliant student, who is currently working in Carl June's group and doing his postdoc, he came up with a cell line that was not responding to the transduction enhancement by PD-16, and it was a K562 erythroid leukemia cell line. When we incubate with this compound, there is no significant enhancement of transduction, and we had a system where we could see the transduction enhancement. So this was a good starting point, and we had the structural analysis and the functional data to identify the putative target. So we did RNA-seq analysis and found that there are, other than FGFR, EGFR1 is highly expressed in the responsive cell versus non-responsive cell, suggesting that it could be putative target that is operating here. So obviously the next step that we did was generated a knockout of EGFR, and we saw that in the knockout cell infectivity increased in the absence of compound even, and the responsiveness to compound disappeared. And we just didn't stop there. We procured other compounds that are known EGFR1 targets, and we found that other compounds were also increasing the transduction levels in the target cell, but they were not as good as PD-16. That means this compound is doing other than what is known for it to in the literature. So to conclude here, EGFR inhibition via small molecules or genetic ablation in this target cell was promoting transduction events in these cell types. Since now we are curiosity driven lab, so since we had a cell line where this compound was not working, and we need lot of cells to be transduced and to understand various aspects of viral replication, so we wanted something as a formulation that works in a universal manner, or at least work in the cell types that we are interested in. So we went back to our screen and identified the events without having those filtering criteria like envelope and all. So we identified some of the compounds other than PD-16 were also enhancing the infectivity. So we came up with a formulation that works in very hard to, and people who work with THP1 knows how difficult it is to transduce with the vectors and get the efficiency. And this is at 1 MOI we are talking about. This cocktail significantly increases from here to here in terms of the number of transduced cells, and we can always get close to 100% if we increase the virus inoculum. But the point is, with the increased inoculum, you have the adverse effect like activation of TLR pathway. VSV is known to induce TLR pathway, and these are immune cells, so they will just fire the inflammation. So this cocktail formulation we checked from the cells of distinct tissue origins like neuronal cells, epithelial cells, lung epithelium, cervical epithelium, monocytes, macrophages derived from the Raji cell line, which is B cell line, and the cell lines that are routinely used, and including the murine leukemia cell line and the K562, where this PD-16, the EGFR target that we identified, was not showing any enhancement with but the cocktail formulation was increasing the transduction efficiency in these cell types. The cell type that is more relevant for this audience is CD34, and we checked in CD34 cell type also whether this cocktail works at 1 MOI, and whether we can actually come up with something that can reduce the cost of cell and gene therapy by reducing the vector inoculum that is being used. And we can see that this cocktail is able to increase the efficiency. However, I want to caution here, because this has almost 4 or 5 different compounds, so it is very important and imperative to check whether it has any adverse effect on the cell physiology. And that's what when we looked at the scatter plots, and this slide is busy, I know, but I want you to focus your attention here, where we see that the scatter of the transduced and untouched cell is something like this. And when we add this cocktail formulation, you can see that the scatter is changing, suggesting that this compound has some impact on either it is inducing differentiation, or it is doing something to the target cell type, which may not be good from the therapeutic point of view. And so we use deductive approach. We started eliminating one by one each compound, and also did analysis by incubating individually this compound with the cell type, and we found this ERK inhibitor that was part of this cocktail formulation was alone responsible for change in the scatter, although it was increasing the transduction efficiency. But it is very important to note that and understand the factors that could be actually determining the cell physiology and cell health. So I think I had too much coffee in the morning, so I finished in time. Looking forward that we are expanding this pipeline. We have the pipeline to expand it to the clinically relevant cell type like CD34 cells, and thanks to Srujan, Sarvana and Mohan from CMC that we joined hand now to expand this program as part of the ICMR Center of Excellence. And we are integrating multiparametric analysis, as we learned from this previous exercise, that we need to check multiple parameters, the cell physiology and cell surface markers, so that these transient manipulations are not even changing those aspects. And obviously we are continuing the molecular mechanism by which this cocktail and the target that we identified is promoting the lentiviral transduction in various cell types. And obviously at this point it is too early to say that it can be used for therapeutic application, and we need more work to focus on the characterization and reducing toxicity. But we are aligning ourselves with the Honorable Prime Minister's mission of Atmanirbhar Bharat, and we would like to contribute to whatever extent we could as a part of developing the enhancer that can reduce the cost of LV that is used for cell and gene therapy application. So I am just here, these are the people on behalf of them I am talking, and they work mostly on these aspects, and this is one of the small aspects that is happening in the lab. There are several other programs, and I am thankful to the funding agency who has been quite generous, in supporting us. These are various funding agencies, and I thank NIBSC UK and NIH AIDS region program for supporting and generously supporting us with the reagents and cell line that we work with, and the collaborators, and I thank you all for your attention.
So thank you for such a wonderful talk, and we can have questions.
Yeah.
Great talk, Ajit. Couple of questions. So, do you see any immune modulation due to these drugs? Is that why you're saying enhance transduction? Is this purely based on entry of virus, or is it also post-translational?
No, I suppose it is not the entry, because that aspect we already ruled out, where we use different pseudotypes which are using different tropism and entry mechanisms. In fact, HIV doesn't require endocytosis. VSVG is dependent on endocytosis, and Chandipura, I think, require to some extent endocytosis. So entry is not something that is the mechanism. It is subsequent post-entry that is helping the virus to vector to transduce better.
So the real question I'm going back to is, if you use these drugs, if they were immune modulatory in some way, if you use these on producer cells, are you going to see a benefit in producer cells?
We checked all those combination, producer cell, time of just due to time constant, I did not add that data. But time of addition, even producer cells with the virus, before incubation, after incubation, all those aspects, and it is basically effect in the target cell. So if you add before or with the virus or after the virus is added, you see these effects. But after the virus, there is a time window like up to 6 hours. After that, because that's the time when virus integrates, right? So 7 hours is the time from the reverse transcription to integration. So if that is missed, then I think it doesn't work. And I suspect it is something to do with the type 3 interferons, but maybe next meeting I will be clear about it.
And do you know about in vivo usage? Is it possible to use them in vivo? Will it enhance transduction?
Because it's not going in vivo, because if you are injecting the cells, then anyway that compound is, transient treatment is there, up to 2 hours you need to treat, you don't need to go beyond 2 hours. So if the cells are being injected, there is no, the compound is not going. But this compound is already used as FGFR inhibitor, so it's already, I think, in some trials. So
thank you.
I think it's not going to be a problem. Yeah.
So I have a question.
Yes, please.
Yeah, sorry.
Wonderful talk. Really enjoyed it. Do you know whether this is specific just to the reporter lentivirus you made, or the cargo it carries influences the degradation? And what percentage of the virus is actually degraded by post-entry before it reverse transcribes and integrates? And how does this inhibition work?
Yeah, so that's a very good question, because whether it is ZS green or the GFP specific that we see, we checked with the luciferase and other reporters, and it is consistent with that. And we see obviously more integration, because the ability to infect cells increases. How? I don't know as of now, but then this compound, what we see is working through EGFR. EGFR itself is now a new target that we found that could be one of the antiviral factors. EGFR itself, or the downstream mechanism that is transduced through EGFR.
So do you see the same, people have used cyclosporin H and all those to repress the restriction factors.
Right.
Do you see any synergy or with those?
We have done with the commercially available retronectin, transdux or something, viroductin, all those, the parallel comparison has been done. So it works, but only for CD34 cell types. We are not yet convinced that this is going to work without inducing that differentiation and toxic effects.
We can have one last question from you.
Yeah. Yeah, we checked several promoters, and it is irrespective of promoter or the coding gene that is being used, it is working. Yeah, I think even experiments are becoming personalized, so we'll have to do CD34 specific screen to identify conditions probably. Yeah.